And here we see the first phase of mitosis called prophase. And as you can see, there are two pairs of centrioles, or two centrosomes, which in the S phase of the cell cycle during interphase, the centrosomes are duplicated. And what they do during prophase is they start to move towards opposite ends of the cell. And the early mitotic spindle starts to form between them, which is made from spindle fibers, which are really thin microtubules, which will be essential to mitosis later in the process. Here we see the second stage of mitosis, called metaphase, in which the chromosomes line up in the middle on the equatorial plate, and as you can see, the centrosomes 1 and 2, or the paracentrioles, whichever you prefer, have gone to either ends of the cell, forming poles, and they have spindle fibers stretching across the cell, which will eventually pull the chromosomes to either ends. They act like magnets, more or less. And now we have come to the third stage of mitosis, which is anaphase, in which the chromosomes split apart and are pulled by the spindle fibers, which are attached to the centrosomes. It should be centrioles right there. Where they are pulled apart to the centrosomes on opposite ends, so that now we have two sets of DNA in one cell ready to be split apart soon to form two new cells. And now we come to the final stage of mitosis, called telophase, in which the spindle fibers, which are made of microtubules, begin to loosen up between the two new cells that are shaping, well, are beginning to take shape right now. And you can see the nuclear membrane is reforming, the chromosomes are recondensing, and the centrioles are on opposite ends of the cells. Now, after mitosis, the centrioles, since these pairs of centrioles and centrosomes, are in charge of nuclear division, they will go into rest after mitosis and just be inactive until they are needed to duplicate again to undergo mitosis again. And here we see that what I was referring to earlier is that centrosomes, which are paracentrioles, are formed of one centriole, can be found in cilia and flagella because both are composed of microtubules. Cilia and flagella provide mobility for most prokaryotic cells, though they can be found in eukaryotic cells as well. Flagella is one long tail that's most likely seen with sperm cells, while cilia are little hair-like structures found around the protist, which help them move through their environment. However, cilia and flagella have microtubule doublets instead of triplets, which have a little bit of a different shape than centrioles, but they're more or less the same thing.